today we discuss about the DNA replication. First of all, what is replication? Replication means the copy of anything. Means here DNA replication means copy of DNA. See, this is the diagram of central dogma. Central dogma is very specific term. That is that contain replication, transcription and translation. These three process. Means here first DNA copy of the DNA, that is called replication, right? We discussed this replication in this video in detail. So first here copy of the DNA, that is replication. Conversion of DNA into RNA means synthesis of RNA. Synthesis of RNA takes place from the DNA. DNA acts as template for the RNA. So this process is known as transcription. And another process, that is translation, that is synthesis of the protein which are coded by the DNA. For that formation of this protein, this RNA will be act as the intermediate. So protein are synthesized from the RNA. RNA act as template for the protein. Then this protein act as a enzyme or other property. Okay. Another here, that is reverse transcription. Means synthesis of DNA from the RNA. Synthesis of DNA from the RNA. This process is known as reverse transcription. So what central dogma contain? What process central dogma contain? Replication, that is copy of the DNA. Another is transcription, that is conversion of DNA into RNA. And another is translation, that is conversion of RNA into a protein. So synthesis of protein from the RNA molecule, that process is known as translation. And another term is reverse transcription, that is take place from the synthesis of the DNA from RNA. This is known as reverse transcription. So first, what is DNA copy? Already know replication means copy of the DNA. So how copy of the DNA would be takes place, where it will takes place and how it can be takes place. Here we study these all things. So in each cell division, cell must copy its entire DNA. Means what happened in cell division? Suppose this is a cell. This is nucleus of the cell. In this nucleus, this contains genetic material, and this is the cytoplasm where all organelles are present, right? After what happens in cell division? In cell division, from one cell, this cell divide into two cells, right? In mitosis. So what happened here? Here for this division from one cell, they make two cells, identical copy. Remember this. So for this identical copy, what happened here? The genetic material and other organelles, that all will be copied here, right? So in this step, what takes place? Replication of the DNA, so that exact copy of this DNA will be present in two cells, right? Cell division having 4 phases, that is G1 phase, S phase, G2 phase, M phase. This is a phase of the cell division. In S phase, replication will be takes place. In G1 phase, all the requirement of the enzyme, protein for the replication, that all will be prepared in G1 phase. After preparation of this protein and enzyme, the genetic material or DNA will be replicated in S1 phase. After replication, all the protein and enzyme required for the mitosis or M phase, mitosis or meiosis, that will be prepared in G2 phase. And after G2 phase, mitosis and meiosis will be takes place. This is M phase. So cell division having 4 phases: G1 phase, S phase, G2 phase and M phase. Okay, clear? So replication will takes place in S phase. S phase in the nucleus, and where? In S phase. S phase of the cell division. Each cell division, cell must copy its entire DNA. So each daughter cell gets a complete copy. Suppose this is a mother cell, then each daughter cell having identical copy of their mother, right? Identical copy of their mother cell. What is the rate of synthesis? In bacteria, 1000 bases are synthesized in per second. In 1 second, 1000 bases synthesized. In mammal, in 1 second, 100 bases are synthesized. Okay? Remember this. In bacteria, in 1 second, 1000 bases will be synthesized, but in mammal, in 1 second, 100 bases will be synthesized. So rate of synthesis in bacteria will be more as compared to mammal. What is the problem in this replication? With a single replication origin in DNA, bacterial genome, the genome of the bacteria is 4 into 10 to the power 6, right? 4 into 10 to the power 6. They takes 20 minutes to copy. Means they takes 20 minutes to replicate their whole genome, because 1000 base will be synthesized per second. Human having 3.2 into 10 to the power 9. 3.2 into 10 to the power 9 genome. So it takes 1000 times longer. 1000 times longer as compared to bacteria. One thing, why in bacteria, in per second, in 1 second, 1000 base will be synthesized? Why? And in mammal, in 1 second, 100 base will be synthesized? Why? This is because in bacteria, the synthesis rate of the synthesis of the base will depend on the origin of the replication. Means opening of the fork, that is called replication fork. Replication fork or origin of replication. This depends on origin of replication. Bacteria having a single origin of replication, whereas in mammal more than one origin of replication is present. But bacteria having only exon part. What? They contain only exon part. Whereas mammal having exon and intron both. Exon and intron both. Means in mammal, coding and non-coding both type of sequence are present. But in bacteria, only coding part will be present. That's why 1000 bases were synthesized in 1 second. Okay? Clear? This is a model proposed in DNA replication. Actually, there are 3 types of model. There are 3 types of model in DNA replication. First is conservative model, second is semi-conservative model, and third is dispersive model. Semi-conservative model is identical model, or this model is used. Okay. In 1950s, three different mechanisms were proposed for the DNA replication, that is conservative model, semi-conservative model and dispersive model. In conservative model, what happened? This DNA is double helix, right? They contain two identical DNA that are antiparallel with each other, right? So this DNA, when replication will takes place in this DNA, the mother DNA will be conserved, means they are separated, and the daughter DNA will be another, they are separated. This is a conservative, means mother DNA is conserved here. Mother DNA will not go with daughter DNA. They act as template for daughter DNA, but they are separated, totally separated, right? In conservative model. In dispersive model, in conservative model, both parental strands stay together after DNA replication. They cannot share with their daughter DNA. They are totally separated in conservative model. Next is dispersive replication. In dispersive replication, what happens? When replication will take place, they combine with their daughter DNA. How? In middle. Like parental and daughter DNA are interspersed in both strands. Means parental and daughter. When they replicate, suppose this is a daughter DNA and this is a parental DNA. Both strands, after replication, both strands having interspersed with the parental and daughter DNA. They are not separated, right? In semi-conservative DNA, these both strands will be separate like this. These both strands will be separated in semi-conservative, and these both strands act as template for the new strand. This is a parental type. After replication, this is a daughter DNA. After replication, this is a daughter DNA. Right? Right? So, in semi-conservative, one strand is parental type and one strand is newly strand, that is daughter. So this is known as semi-conservative. They are not totally conservative. Means in conservative, what happened? Parental strand is separated and daughter strand is separated. Both are stay alone. Both strand of parent will be stay together and both strand of daughter will be stay together. But in semi-conservative, what happened? One strand is daughter DNA and one strand is parental DNA. Right? So, this semi-conservative model of the DNA will be accepted. What? This is accepted model. Semi-conservative. Okay. Clear? There are three types of model: conservative, dispersive, semi-conservative. In conservative, both parental strands stay together, right? In semi-conservative, one strand is parental type and one strand is daughter. So, this is known as semi-conservative. In dispersive, parental and daughter DNA are interspersed in both strands. after replication. Okay. See, this is a overview of the replication of semi-conservative. Okay. See, this yellow part is a parental DNA. What? This is a parental DNA. After replication, one strand is parental type and one strand is daughter. This yellow type is a parental strand. This is a parental strand. This red is a daughter. And again after replication, one strand is parental and one is a daughter strand. Right? So this is a process of semi-conservative model. Semi-conservative, this contains semi-conservative model. Right? So, another is what happened in DNA replication. What happened here? Double helix strand. What happened? DNA is a double helix strand, right? This is a DNA. DNA is a double helix strand. This both strand will be copied. Means replication will be takes place of this both strand. Semi-conservative solution, each daughter cell get one of their original copy. Unwind at one point and use that as the origin of replication. Means they unwind. Means this is a double helix DNA. The nucleic acid are attached with hydrogen bonding. Helicase came and they break this hydrogen bond, and this strand will what happen? This strand will be separated. Means this is a separated strand, and this is act as a origin of replication. Okay. The enzyme, there are several enzymes that are used in the DNA replication. What are that enzyme that are used in DNA replication? The main enzyme is DNA polymerase. What? DNA polymerase, main enzyme. Actually, all enzymes are main enzyme. Any lacking of any of them, this enzyme, the replication would not take place, or mutation will be happen. Okay. So what happened here? First is DNA polymerase. DNA polymerase 3. There are 3 types of DNA polymerase, 1, 2 and 3. In replication, main is DNA polymerase 3 that polymerize the DNA template. Okay. This is the DNA polymerase enzyme. Function, they replicate enzyme. Means they polymerize the DNA. Second is DNA polymerase 1. They help in repairing. If any mutation or any mispairing of the bases will be takes place, then they go back means they go in 3' to 5' direction. Replication will be always takes place from the 5' to 3' direction. So repairing will be takes place from the 3' to 5' direction. 3' to 5' direction. So DNA polymerase 1, they act as repairing enzyme, and they repair the mispairing or mutation that occur during the replication in 3' to 5' direction. A repair enzyme involving removing error and filling in gap in the sequence. DNA ligase, what they do? DNA ligase means sealing. Means they seal the gap or nick. Seal the gap or nick that occur after the removing of the primase, after the removing of the primer, or removing of the nucleotide. They seal that gap in the sugar phosphate backbone. Where? In sugar phosphate backbone. Next is RNA polymerase. You think that what happened here? What happened? What is the use of the RNA polymerase in DNA replication? Here is the use of the RNA polymerase, because DNA replication will be not takes place without RNA primer. What? Without RNA attachment of the RNA primer. Suppose this is a DNA, right? This is a DNA double strand. When this is 3' to 5', so replication will be takes place from the here, right? So during the replication, here primase will become, primase will be attached here. They synthesize the RNA primer. This is the RNA primer. RNA primer. For the synthesis of this RNA primer, RNA polymerase will be required. Right? RNA polymerase will be required. They synthesize the 10 to 15 nucleotide of the RNA primer. After that DNA polymerase, this DNA polymerase come and bind with this, and the replication will be initiated. Okay. So RNA polymerase or RNA primase, both are same. They make RNA primer to get the DNA polymerase 3 started. Right? Understand? Clear this? Next is RNA-H. What is the function of RNA-H? They remove the RNA primer. They remove this RNA primer. What? They remove this RNA primer after the replication, when replication will be completed. They remove the RNA primer once they have completed their function. DNA helicase. DNA helicase, they unwind the DNA, or they break the hydrogen bond. What? They break the hydrogen bond. Another is DNA guidase. DNA both having same function. DNA helicase break the hydrogen bond, and DNA guidase or topoisomerase both are same. They unwind the DNA double helix. Twist or unwind the DNA double helix. Another is helix destabilizing protein. Helix destabilizing protein. They prevent the unwound DNA from immediately rewound. That is SSNP, single stranded nucleotide for protein. They go and attach with the single stranded DNA so that they cannot bind again. Okay. These are the enzymes that are used in DNA replication. Right? First is DNA polymerase 3, DNA polymerase 1, DNA ligase, RNA polymerase, RNA primase, RNase H, DNA helicase, DNA guidase, helix destabilizing protein, that is SSNP. This is the diagram of DNA replication, how the replication fork will be opened. This all are here. See, there are two strands actually, template and non-template. This is a double helix strand. Okay, this is a double helix, one helix that act as template. This is another helix. Okay. So already I told you that replication will be takes place in the 5' to 3' direction. So this is a template. What? This is a template strand. Here, what happened here? See, this is a 5', right? Means replication will be takes place where? Replication will be takes place from where? In opposite direction, right? Right? This is a 3', right? This is a 3'. Means this is replication will be takes place from here only. So what happened here? In this direction, first hydrogen helicase came and break the hydrogen bond, right? Helicase enzyme, this is a helicase enzyme, they break the hydrogen bond, right? After that, topoisomerase came, topoisomerase or gyrase enzyme came, right? They unwind the DNA, okay? After that, SSNP, single stranded binding protein came and attached with this, so that they cannot form a helix. They cannot form a helix again, okay? So this is a single stranded binding protein. Go and bind with this single stranded, right? After that primase came and attached with this. Primase came, this is a primase, right? This is a primase. Primase came and form a primer. This is a primer. After formation of primer, DNA polymerase come and bind with this, right? This is a DNA polymerase and bind with this primer, right? After binding with this primer, they continue the synthesis. But remember, this is a lagging strand, means they act as opposite strand, right? They act as opposite. So in this situation, what happened? Short nucleotide of the base will be formed. They remove the, again primer will be removed. Again another short nucleotide of this strand will be formed. So here a Okazaki fragment will be formed. Means there are several formation of short strand will be takes place. So this strand is known as lagging strand, or Okazaki. This fragment is known as small fragment is known as Okazaki fragment. What? A small fragment. A small fragment is known as Okazaki fragment. Okay? Clear? Because this is happen because they move in opposite direction. Okay? Fork will be go in this direction. The replication will be takes place in this direction. So this will be happen because DNA polymerase synthesize only the 5' to 3' direction. That's why this will be happen. So in another strand, what happened? This is a leading strand. In this direction, in the direction of the replication fork, the polymerase will be moved. So here only one strand will be synthesized. Complete one strand will be synthesized here. So this is known as leading strand. After the synthesis of this Okazaki fragment, what happened? DNA polymerase 1. What? DNA pol 1 came and bind with this. They move in 3' to 5' direction. They move in the 3' to 5' direction and remove all the mispairing of the nucleotide and remove the primer and remove the RNA primer. After the removing of the RNA primer, gap will be formed, right? Gap will be formed. Then what happened for this gap? Another enzyme, that is DNA ligase. DNA ligase came and bind with this gap and they seal the nick or gap. So here also one single strand will be formed. In leading strand, this all process will not take place, but in lagging strand this will be takes place. DNA pol will came, they remove the primer, then DNA ligase will came, they seal the gap, and formation after that formation of one single strand will be takes place. So this is the process of the DNA replication. what happened here? See, this is a strand, right? This is the replication origin. What? Origin of the replication. DNA helicase and topoisomerase came. DNA helicase and topoisomerase. What? Topoisomerase came and bind with this. They break the hydrogen bond. They break the hydrogen bond here, and topoisomerase unwind the double helix. Then this is known as fork. What? Fork. This is known as fork. And in this direction where fork will be moved, in that direction only polymerase will be moved. If they not move in direction of the fork, if polymerase will not move in the direction of the fork, then Okazaki fragment will be formed. What? Okazaki fragment will be formed there. This is the actual. When we see the cell division in cell from microscope, you see this figure only. See, this is a microscopic diagram. This is a microscopic diagram. This is the DNA where replication will be takes place. This is the replication, right? This is the replication where replication will be takes place. So from microscope you see this diagram, how the replication will be takes place actually, right? This is the nucleotide base pairing. How the nucleotide will be base pair when replication will be takes place. This is the template strand, and this is this incoming or daughter strand, right? So what happened here? From 5' to 3', this is the direction of the polymerase movement, because polymerase move from 5' to 3' direction, polymerase 3, right? So when they move in this direction, the nucleotide according to this template nucleotide, their base pairing for their opposite nucleotide. Suppose here is A, then here T will be came. T nucleotide will be came and bind with this by hydrogen bond, by the formation of hydrogen bonding. So according that only new strand will be synthesized. Synthesis of new strand will be takes place, right? See, DNA replication. The DNA is copied by a replication machine that travel along each replication fork, right? They move in the direction of replication fork. Means this is a replication fork, this is a replication fork, then this is move in this direction. One of the most important member of this complex is DNA polymerase. Already I told you, DNA polymerase. That is the main enzyme that polymerizes the whole DNA. It is able to add DNA subunit making new strand. Means they add the nucleotide. What? Subunit means they add the nucleotide. Adjacent nucleotide, complementary nucleotide of the template strand. DNA polymerase add the complementary nucleotide of their template strand. DNA polymerase first discovered by Kornberg. He is the scientist who discovered the DNA polymerase in 1956. First in bacteria, that is E. coli. There are 3 types of DNA polymerase, already I told, 1, 2 and 3. 3 is the main replication of the DNA polymerase. 1 is help in repairing, 2 is also help in repairing. Human having 4 types, that is alpha, beta, delta, right? But in mitochondria, gamma. Remember this, mitochondria having DNA pole gamma. This gamma synthesized only, help in replication only in mitochondrial DNA. Remember, always question will be asked. In mitochondria, which DNA pole will be required for replication? They given option alpha, beta, delta, gamma. Right option is gamma, right? Don't confuse with gamma. Gamma are not used elsewhere except mitochondria. Okay, remember this. So all DNA pole have two properties. They synthesize DNA in one direction. Already I told you, that is direction is 5 prime to 3 prime direction, and they add and only add to the end of existing double stranded DNA. They cannot start synthesis of DNA from scratch. RNA polymerase can, but not DNA polymerase, right? Means DNA polymerase require RNA polymerase to synthesize the replication, because DNA polymerase only replicate the DNA when primer will be present, and that primer will be synthesized by which enzyme? RNA primase or RNA polymerase. This is the replication fork, right? Already you understand this. This is the replication fork. After the breaking of the hydrogen bond, this is the direction where replication fork will be moved, right? One strand is the leading strand, act as a leading strand, one is lagging strand. This is the leading strand, this is the leading strand, act as leading strand, and this act as a lagging strand. After that, DNA pole 2 will become, they remove all the primer. After that, ligase will become, and they seal the nick, right? So here also one strand will be formed. What happened at the other strand? 3 prime to 5 prime strand replication solved by Rezi Okazaki. He saw one strand made in a continuous manner, right? Means they act as a leading strand, and another is short discontinuous pieces, that is the lagging strand. That discontinuous pieces, already I told you what called they called as a Okazaki fragment, right? They called as Okazaki fragment. This is a template strand, right? DNA template initiated by RNA synthesis. RNA polymerase will be came and synthesize a small 10 to 15 strand of the RNA primer. RNA primer will be added here, right? Right? Another DNA polymerase came and bind with this RNA primer, right? Synthesis of RNA primer will take place. Again, DNA polymerase come and bind with this RNA primer, then they synthesize DNA. So RNA is added by primase, they synthesize 3 to 10 bases. DNA is added by DNA pole alpha, right? Primase can start RNA synthesis. We have a RNA DNA joint, so RNA is involved in eukaryotic DNA replication, right? Clear? Closing the gap between Okazaki fragment. Which enzyme close the gap? That is DNA ligase. DNA ligase close the gap. In prokaryotes, DNA polymerase 2 came and repair the Okazaki fragment. In eukaryotes, delta will fill the gap. Which delta will fill the gap? This is the delta. RNase H come and remove the RNA primer, and DNA pole delta fill the gap. DNA ligase gap closer, right? How the stabilizing of the replication machine? DNA pole delta and epsilon are attached to the DNA. This will be happening in eukaryotes. DNA pole delta and epsilon. Pole delta and epsilon are attached to the DNA and held in a place by other protein. Sliding clamp protein, that is proliferating cell nuclear antigen, that is in short form they call it PCNA, allow the stable binding of DNA pole and strand synthesis. Clamp loading protein in attaching the sliding clamp protein, right? Means in eukaryotes, all the replication process will be takes place by clamp loading protein. Okay. This is the diagram of clamp pCNA. This is a pCNA, that is a clamp loading protein, and in this pCNA RFC all will takes place. They move in the direction and load and they synthesize the DNA replication. Polymerase came and bind with this clamp loading protein, and they synthesize the nuclear strand of the DNA. So replication will be takes place in this direction in eukaryotes. Has a proofreading machine built in, check for base machine. This is the function of DNA wall. Remove mismatching bases by going backward. Reason why it is not able to build DNA in the 3' to 5' direction. No energy from ATP hydrolysis. In remember one thing, that in replication energy will be not required. Make just one error in 10 to the power 8 or 10 to the power 9 bases added. Okay. Here, this is already you know what happened for topoisomerase. They prevent the binding of the double helix, or they untwist the DNA helicase. There are two type of topoisomerase, 1 and 2. One break one strand only, and 2 break both strand, and they rejoin. And they single stranded binding protein. They help in binding or making the helix of the double stranded DNA, right? This is the double helix DNA. Already you know this is replication for leading strand, lagging strand. Okay. This is the diagram of all the replication process, what happened, how they work. This is the topoisomerase enzyme, right? This is the topoisomerase enzyme. They go and bind with, they unwind the DNA. After that helicase will come. They break the hydrogen bond. Primase will become. Then RNA primer will be synthesized. After that DNA SSNP will become. SSNP bind with this single stranded DNA. Polymerase bind with RNA primer. DNA polymerase 3. Then synthesis of DNA will be takes place. Right? What is the telomere? Actually, the end of the DNA, that is known as telomere. For telomere synthesis, another enzyme is present, that is telomerase, and they act as reverse transcriptase. Okay. This is the summary of the DNA replication. What happened in DNA replication? This is the first helicase unwind the double helix DNA. Second, single stranded protein and bind with the single stranded protein bind with the parental strand. This is the third step. The leading strand is synthesized continuously in 5' to 3' direction. Another is 4, that is lagging strand will be synthesized. Last is 5th, that is RNA primer will be replaced by DNA. And in Okazaki, DNA ligase will come and join the fragment. So, this is the complete video of the replication. Okay. This is the difference between prokaryotic and eukaryotic DNA replication. In prokaryotes DNA replication will occur in cytoplasm. In eukaryotes occur in nucleus. In prokaryotes, only one origin of replication will be present. In eukaryotes, more than one or many origin of replication will be present. Origin of replication is about 1200 or more nucleotide in length. In prokaryotes, they are having about 150 nucleotide. Replication occur at one point in each chromosome. In eukaryotes, they occur at several points. Only one replication fork will be formed. Multiple replication fork will be forming. Already I told you, multiple replication fork will be forming eukaryotic DNA. Initiation is carried out by protein DNA A and DNA B. Initiation is carried out by recognizing complex. DNA guidance is needed, DNA guidance is not needed. Okay. These all are the difference between DNA replication in eukaryotes and prokaryotes. So this is all about the DNA replication. So thank you.